Okay, well, let's get started today. We've got a little bit of catching up to do. I just want to kind of make sure everyone's feeling comfortable with where we are. Last week, of course, we didn't have any labs, and I just had a lab this afternoon, and I feel actually that the students did pretty well with learning the content over the week, so I'm pretty pleased with how I'm seeing last week going so far. Last Monday, of course, was a holiday, so there was no lecture. Last Wednesday, there was a lecture for you to watch on YouTube, and I hope that you're feeling reasonably comfortable. In that conversation, we finished chemistry, right? And that's really my big concern today, is making sure you understand what's important regarding the chemistry, because that's on the test on Wednesday. So you've got a test on Wednesday. That test is over Chapter 1. And what is Chapter 1 all about? Body regions, introductory terms. It was the atom-to-atom continuum. What is anatomy? What is physiology? What is homeostasis? That conversation. Imaging at the end. What's a PET scan? What's an MRI? What's a CT? That kind of thing. Then Chapter 2 was everything from atoms and molecules, macromolecules, and that's where I also went through and described to you the functions and different examples of carbohydrates, proteins, and nucleic acids and lipids. Now that was a very basic level, but again, for some people that's brand new material. So that's where I want to pause right now and see if there's anything in that conversation. So we're in the Chapter 2 notes, and I can pull those up right now. Is there anything in there that you are feeling uncomfortable about that you want to confirm? The rest of the conversation on cells and all that, we can wait a little bit on that. That's been going on in the background, and that'll be on Chapter 2 exam. Then the last thing recall that on this exam will be the material from Lab 1. And that was also body systems and directional terms and areas of the body and serous membranes and regions of the abdominal pelvic cavity, those types of things. So let me just back off and just ask for any questions at all related to the chemistry, especially the macromolecules, because we didn't get to talk about that face-to-face. And specifically, we're looking at we got through all the electron shells, we got through ionic and covalent bonds, and I believe we got through pH, yes, individually as a group. And then after that is where we picked up the organic compounds, and I think, how far did I get? Did I get it live? I got through metabolism, I got through, I think, through the carbohydrates, right? And then the lipids, the proteins, and nucleic acids for you to pick up from the online section. So what do you think? What in there is confusing or needs to be clarified? Yes, sir?
The vocab for the exam 1.
Vocab for the exam, first 20 slides. So that's about 80 terms. Okay, so not the first 20 terms, but the first 20 slides. On each slide, there's 4 or 5 terms. And that vocabulary is mostly fill in the blank. So it is something you need to know the terms. And recall, I gave you an example on the first day or the second day saying, if you know what ambi and what algia mean, then on the exam I would put those two together. What does ambi-algia mean? And for 2 points you would tell me both sides painful condition. And I will ask that you write them in the order that I present the terms. And so I'll tell you that on the exam. But if I put ambi-algia, I would want you to put both sides painful condition, because then I know that you know ambi means both sides and that algia means painful condition. But that'll be very clear on the exam.
Can you kind of go over just a little bit more about these, like how they break down?
Okay, so I think we're looking at a question on the 3 different ways to think about chemical reactions.
Yes.
So let me go to that right now. So this would be some of Chapter 2, and the concern is somewhere around here. So this is, what page is this on, anybody?
34.
Page 34 of your notes. And this is just an example. Again, you're not responsible for the actual structures here. But what you're looking at are 2 monosaccharides on the left. And these monosaccharides, it tells us, we got glucose and glucose. So we have glucose plus, there's a plus sign missing, glucose plus glucose. And under a chemical reaction, arrow, these 2 single monosaccharide molecules can be linked chemically by a covalent bond to form maltose, which is considered a disaccharide because it's two simple sugars attached. And here we see that there's a covalent bond, not to get too fancy about that. And what does that covalent bond means what? That the electrons are now being shared. And as a result of this making of maltose, a water has been released. That going from left to right, in the order in which I just described it, okay, going from left to right, there's three ways we can describe that reaction. Number one, we're synthesizing something. So it's a synthesis reaction. We're building. Number two, because we're building, we can also call it an anabolic reaction. Remember, anabolic versus catabolic. Anabolic is building. And specifically, this is also called a condensation reaction, because what do we get out of this reaction? Water. And when you're boiling your spaghetti and water condenses on the side of the pot, what do you get? Water. So we get water out of this thing, so that's a condensation reaction. All three of those terms can be used to describe this thing going from the left to the right. If we turn it around, right, molecules in our body are also broken down. So if instead we started out with now maltose, okay? It's a disaccharide. Now we're going to be going from right to left, arrow going to the left. Another enzyme's involved here. Now, I never said that, but in your body, almost every chemical reaction that occurs in your body is under the control of an enzyme. And an enzyme is a biological catalyst without which the reaction probably would not occur. Very, very much speeds it up. So we've got a chemical reaction. And what happens going from right to left? We're breaking it down. So we can call it a decomposition reaction, catabolic reaction, and in order for this to happen, we had to break this water molecule down and redistribute it on the two sugar molecules. So this is also called a hydrolysis reaction. Think hydrolysis, the breakdown of water. They're all, in my mind, they're all going to be equivalent. There are different ways of thinking about the same the same equation. Okay? Question? Nope? Okay. Okay. Does that help? Does that clarify?
A little bit.
Okay. So again, you never get to draw these out, recognize them, just simply know anabolic and synthesis and condensation belong to the same kind of equations, and then hydrolysis, catabolic, and decomp are on the other side.
I have a question actually about one of the things that we did on our mastering.
Another thing on mastering, sure,
Or,
absolutely.
like, from you had like a chemical bonded thing, and you had to figure out how to break them down. What went on one side to the other side?
More of a balanced equation?
Yeah, I had
Yeah.
a hard time with this. Is that gonna be on the
That will not be on the exam. Okay. Now let me relieve your concerns a little bit. The exam, 98% of it are things that I have directly discussed. Okay. So the exams will be directly out of what I've discussed on the PowerPoints and in the presentations. Mastering is there to help you. If it's something in mastering that's directly like what we did in lecture, then sure. But here's what mastering does, just so all of you understand this. Probably that reaction is not one, that problem that she's describing where she had to balance an equation is not one that I probably assigned. Okay, it's probably in the adaptive follow-up. Okay, here's what, let me just explain adaptive follow-ups. So you took the Chapter 2 homework assignment and it decided that you didn't know much about chemical bonds. So all of a sudden it says, okay, she needs to know about this in addition. And it may throw something at you that isn't something we've directly done here in class, but the computer, right, senses that's something that would help you better understand this. Now, because, and chemistry is the one area where it's the worst, because they're pulling stuff over from places that I'm not going to and not expecting you to know. Later on in the course, when it's more, quote, pure anatomy and physiology, then those adaptive follow-ups will be pretty much anatomy and physiology things. But in the chemistry unit, it's kind of notorious for grabbing things that aren't right in front of you and that important. So you were a little lost. That's okay. And that's what chemistry is all about, being lost. That's the goal, I think, of chemistry. So that's, just be relieved on that. Okay. Be relieved on that. Okay, other questions, comments about what are lipids? Give me some examples of lipids. Cholesterol, fats, oils, right? Those are all lipids. Phospholipids, right? The major lipid of your cell membranes. Did I hear steroids? I don't think I did. Steroids are also lipids, right? Testosterone, estrogen. Those are fat molecules. They're hormones, but they're They're made up of, they're steroidal hormones. Then how about nucleic acids? DNA and RNA. I just briefly mentioned the double helix. What's the difference between DNA and RNA? DNA contains what 4 letters? A, C, T, and G, whereas RNA doesn't have T's, instead substitutes U's. That's a big one. RNA is usually double-stranded. DNA is usually double-stranded. RNA is usually single-stranded. Ta-da, we're done with RNA and DNA, honestly. What letters go together? C's go with G's and A's go with T's. That's in DNA. If you see U mentioned anywhere, it must be an RNA molecule. Now, we'll come back and talk a little bit more about DNA and RNA next chapter, but for test number 1, right, content from chapter 2, that's about it. And then what about proteins? Proteins are composed of amino acids, of which there are how many of them? 20. And those 20 amino acids can link together, and we call that linking together the primary structure, right? One amino acid, then another amino acid, then another amino acid. Kind of looks like a train, right? A bunch of cars in the train. But what does that protein do? It doesn't stay as a straight line. Those amino acids do what? They roll over and ball over on each other, and they make a secondary and tertiary structure. The tertiary structure is the third level of structure, and that's the lock and key idea. So every protein has a particular structure, and that structure is related to its function, right? So every protein folds in a unique way to do its job, and if we heat up a protein, what happens to it? It unravels, right? And the word for that is it denatures. And that happens not only at high temperature, but also at low pH. So when you consume protein, when you eat in your diet, and that food gets down into your gut, into your stomach, in the low pH of your stomach acid, those proteins do what? Start to unravel, and that makes them easier now for other enzymes to come in and chop it up so that those proteins get broken down into their amino acids, the sugars get broken down into their individual sugar groups, and every other molecule you consume is being broken down. So the pH of the stomach kind of denatures everything, and then other enzymes come in and continue to chop it up. And then where are those smaller molecules absorbed? We leave the stomach, we go into the small intestine, and that's where those molecules that have been chopped up are now absorbed into your bloodstream. We'll talk more about that, but that's really the basic big story idea that I want you to get out of chapter 2 when it comes to chemistry. Okay, just know examples of them, know what examples of steroids are, what they do, high energy, right? Fats are high energy, those sorts of things. Question?
What is the difference between pinocytosis and exocytosis?
Okay, so pinocytosis and exocytosis are actually going to be in the next chapter. So I will entertain that in a minute, but I'm not going to worry about that. That's going into chapter 3. Right now I'm focused on chapter 1 and 2. Anything from lab for the exam on Wednesday, okay? Anyone who's listening in, hybrid students, as you saw in the email, you are to call or contact the testing center. You can do that through MyMCC, Student Services, or just call them and schedule the exam for Wednesday. The exam will be here at 4 o'clock. Anyone who's in hybrid is welcome to join us here at 4 o'clock or schedule earlier. Keep in mind, the hybrid group has lab on Wednesday. So they have lab from 12 to 3:45. So they can either come at 4 and take it with us or take it earlier in the morning. Most of the other exams in this semester are on Monday. So I think this is the only Wednesday exam. So anyway, hybrid students, you got to deal with a lab and a lecture exam on the same day. Okay, any other concerns? Are we feeling comfortable? I don't sense you're too stressed out. Feeling pretty reasonable? Okay. Tonight was the deadline, was it not, for the quiz? Yes. Okay, so make sure you do the mastering quiz. And I sent out my little slap on the hand email. I don't know if it's you guys, I didn't compare notes and names, but as I go back and look at the assignments over the last week, there's always been 10 to 15 students who have not done them. And I don't know if it's always the same 10 or 15 students. I haven't looked at that carefully, but those assignments are important, and they're going to help you be prepared for the exam. Repetition, repetition, repetition is important in this course. And at the end, if you haven't done those assignments, it's going to hurt you. Not only were you not prepared, but you will be losing points on your mastering point. So please, those are important. If you miss 1 or 2, it's okay in the end. Just don't make a habit of it. So that's my little announcement on mastering, all right? So just don't let it slip by. Check it out, check the calendar. I've already loaded the assignments for exam 2. They're not all visible to you yet, but they're all there. So as soon as the exam is over on Wednesday, you can certainly look at mastering and get an idea of what's coming up. There'll be 2 or 3 homework assignments and a quiz. It's not major, but that's what's coming up for exam 2. Okay. Anything else on exam 1? Another thing I will suggest to you, if the chemistry part is, if you're struggling with that, and a lot of students do, on this test, the weakest little area is the chemistry, because that's the thing that's least familiar for many of you. If you listen to the lecture again, so maybe just pop on YouTube and listen to that chemistry section of the lecture again about protons and electrons. If that's a conversation that you struggle with, and you know that's you, fine. Go listen to it again. And you can do that. That's nice about this. And also pre-lab number 2. So you know that in pre-lab number 2, there was that conversation about water and sodium chloride. Those were the same molecules that I talked to you about in lecture, same ones you had the pre-lab activities to do on, and the same ones you'll be tested on. So as long as on the test you know that a covalent bond, a good example of that is water, polar covalent, that you know sodium chloride is an ionic bond, you'll be okay. It's the same story. Okay. So hopefully, even if you're not really, really super comfortable with it, you'll learn it well enough for the test, and then it will sink in. Okay? It is not, I'm not trying to make it trivial, but it's important. Okay, anything else from the chapter? I'm kind of thinking not to do vocabulary today, because the online lecture went through 20, did it not? Yes, right? And so I may not do vocabulary with you today, right? So that next time we'll just get going really hard when we get back on the vocab. Please know that 1 through 20, what is it, A through cusp, is on the exam. When we're done with the exam on Wednesday, you can certainly go ahead and make up the next note cards. It'll be 21 through 42, I think it is, whatever the syllabus says for the second exam. But I won't go ahead just so I'm not adding more vocabulary to your head, so you don't have to worry about that today. Okay. And I'm going to pick up with the second half of chapter 3. Okay. Now, so I'm going to skip over vocabulary. I don't do that very often, but I think that'll be worthwhile today. And then if anything, as I go through any of this, I want you to jump on me and tell me if something needs to, if something, you're reminded of something that you want to go back and look at. So I'm going to pick up with, let me get this open. This lecture doesn't want to open up. Hold on. That's strange. It just doesn't want to open up. Okay, there we go. Okay, so here's chapter 3. Now, again, this is the content that is on exam number 2. I'm glad you're here. It'll help you that you're here, but what you're hearing right now is not on the exam on Wednesday. Let's go ahead, though, and just remind, let's do this really quick. So when you look at image A, what plane was used? This is good for the exam. What plane is used for image A? Sagittal. Maybe mid-sagittal, right? But at least it's sagittal, no doubt about that. And then what about B? Frontal, also called coronal. Either word's fine. And C? Transverse, also called
Horizontal.
cross-section more often. Horizontal, rarely. I don't use that word in my everyday language, but cross-section or transverse. Those are the two you'll hear the most commonly. Let me just dim this light in the front just a little bit. Is that okay? Is that better? That's too dark. Okay. A little bit better. Now, I know that you're not going to be playing any kind of catch-up or worried about this. Your primary focus over the next couple of days is exam number 1. But before I see you a week from now, right? It's going to be a whole week before we have new content. So today, I'm talking on chapter 3. And chapter 3 is a big one. There's all the cell organelles and the parts of the cell, and then it goes into transcription and translation. And transcription and translation, I always think about it, are the fun things that cells do. So this whole chapter is cells. It's all about cells. Chapter 4 will be histology. Chapter 5 will be skin. So that's where we're going through our continuum today, finishing up cells. Next week, starting on histology. You're doing histology in lab this week as well. And then we'll start on skin, which is also just touched on in tonight's lab, or today's lab. So that's our madness. The second exam will be chapters 3, 4, and 5. It'll be cells, histology, and skin. That'll make up exam number 2, and that's still a couple weeks away, but it's not bad to get our minds around what's going on here. So where I wanted you to listen to on the YouTube video, and if you haven't had a chance to do it, these 2 conversations I'm about to have with you are kind of 2 different pieces. Definitely pick up and listen to the cells, okay, the cell talk on organelles. Who had a chance to listen to that? Some of it. Now, I had one person email me and said that the second link was not showing for the video to watch. Did anybody else have that issue? On YouTube, did it say, watch this video, start at minute 40, and also watch this video? And there was nothing there? And also was blank? Okay. It was there. Somehow it was hiding. So you may not have watched that second half, okay? But I need you to go back and do that. And you got a whole week to do this. So this chapter 3 has 2 parts. It's the cell. What is a cell? Parts of a cell, organelles, Golgi, endoplasmic reticulum, nucleus, all of that conversation. And as you're going through this talk, the biggest thing that I'm going to be spending time on in here is the membrane. Okay? So you go back and listen to that. And at the end of that presentation, it stopped at, it kind of stopped strangely, didn't it? At the very, very end of the presentation that I gave you. So that's where I'm going to go, and I'm going to pick up with that conversation. So you're going to review the cells and the organelles. Oh, let's just do it really quick. So this is a typical cell, actually an atypical cell. This cell would never exist on the planet. There's some things on this cell that would never coexist. But I'll just point, and we'll just do this as a quick review, and then you can go back and listen to it. So I'm going to point to some different organelles, and you tell me what they are, if you know. And the real big take-home lesson from this whole first part of the chapter is, can you name the organelle and what does it do? That's the big thing. Can you name the organelle and do you know what it does? So we'll start on the outside. So outside, all of this is the what? Plasma membrane or the cell membrane. Coming off from the membrane, there can be 2 or 3 different appendages. These are cilia. They're going to be found in the respiratory tract, for example. They're going to be found along the fallopian tube to help push the egg along. That's where you're going to find cilia. There can also be, never on the same cell, little tiny finger-like extensions. They're shown all over the side of that cell. Those are microvilli. Microvilli are going to be there to increase surface area. You're going to see them lining your intestinal cells and increasing surface area for absorption of nutrients. And this picture doesn't show it, but the other appendage would be potentially a sperm, or on a sperm, right? It would be the flagellum. So again, they would never all coexist, but that'd be the third and final type of appendage that a cell could have. Now, once we're inside the cell, we got past the plasma membrane, then we have these, in this cartoon, these orangey-looking beans. These are mitochondria. Now, what are the mitochondria doing? They're making ATP, right? They're the powerhouse of the cell. They're making the energy necessary to run all the other processes. And what is this green ribbon candy looking thing? Golgi apparatus. Now, the Golgi is going to be, quote, packaging. It is the packaging center, the UPS, the FedEx center of the cell, and it's going to receive the proteins and molecules made by the cell and package them in unique ways. Then we have right here those little, looks like some little Lincoln Log toys. Those are the, collectively, it's the centrosome. It makes up both parts, and each of those individual little tinker toys is a centriole. So ol, as an ending, ol means small, ol. Arterioles are small arteries, centrioles small. So the smaller part, the more individual part is the centriole, and together they make up a centrosome, zome meaning body. Now they're going to be very, very important in a process you've learned about, mitosis. So those centrioles are going to be important, and they're going to spit out the microtubules from which the cells are going to pull the chromosomes apart and divide. Right next to the nucleus is a series of membranes, and those membranes have little dots on them called ribosomes, and that membrane would be the rough endoplasmic reticulum because there are ribosomes on it, and endo, right, within, plasma, it's a bunch of membranes, plasma membranes, reticulum meaning network. So this is a network, a series, a grouping of membranes inside the cell. And then some of this endoplasmic reticulum is lacking ribosomes. Kind of hard to tell here, but these guys are a little further away from the nucleus and do not have ribosomes attached to them. That is the smooth ER. Now, the smooth ER has a different job. It doesn't have ribosomes, so it's not making proteins. Instead, what's made at the smooth ER? Lipids and steroids. Okay, so the proteins are made on the rough ER with the ribosomes, and the smooth ER is responsible for making in your body some of your lipids and steroids. Then we move to the nucleus itself, right? The control center of the cell. That's where the DNA is found. We don't see the DNA in there, do we? We don't see the chromosomes. So if we don't see the chromosomes, what stage of the cell cycle are we in? I can't see chromosomes. Interphase, right? Interphase is the time during which the cell is not dividing and during which you would see the nucleus and you would not see the chromosomes. So that's consistent here. I don't see any chromosomes in there. They're kind of hiding from me. And also, if this were anywhere near metaphase, if this was anywhere near mitosis, where would those centrioles be? On opposite sides, right? They'd be ready to spit out their microtubules and pull the chromosomes apart. So I got a couple different ways of thinking about this cell. Certainly not in mitosis. It's definitely somewhere in interphase. The very center of the cell, in the very center of the nucleus, there's a darker staining area called the nucleolus or the nucleolus. You'll hear it pronounced both ways. And the nucleolus is responsible for helping to compose the ribosomes. Okay? So the ribosomes are little protein synthetic nuggets that we'll talk about, and they are made in part in the nucleolus. So that's a really quick rundown through this. The last slide of this chapter also has a quick review slide on that, and that's what I want you to do as you go through this presentation. As you go through this presentation, what you're going to note is that it is basically a slide or two on each of the organelles, right? So go through the nucleus, go through the membrane, and this will walk you through this. Now, again, what you're focusing on is going to be, can you recognize the organelle, and do you know what the organelle does? I spent a lot of time in this presentation talking about the plasma membrane. The plasma membrane, of course, is the divider, right, between the inside and the outside of every cell in your body. You've got all these trillions of cells. They need to get the nutrients and the oxygen in, and they have to get waste products out. And so this is a very important dynamic going on at the cell membrane. So I spent a lot of time in this presentation looking at that. And in lab last week, there was a little bit of mention about solubility, how like dissolves like, and how some molecules can get through or interact with fat layers. So that'll be an important conversation. So before I see you again, if you didn't already do this, make sure you listen to this first half. On Blackboard, it says Lecture 3. Okay, Lecture 3. And that's the one to make up from last week. So there seemed to be a piece of that missing. Okay. So passive transport, pinocytosis, and exocytosis. That was a question you had. I'll go ahead and address it. The question was, what was your question? You were asking right beforehand. Okay, the difference between exocytosis. Okay, there are 2 or 3 different types of exocytosis or endocytosis. These are ways that things get into and out of the cell. The 2 different main types of endocytosis, that is things coming into the cell. One is phagocytosis, one is pinocytosis. Okay, those are things going in. Exo is things leaving. Okay, so pinocytosis, pino means drink. So it's slurping from the environment, small molecules. Phagocytosis, phage means to eat. So these are large particles that are being enveloped and sort of bear hugged and brought into the cell. Those are both examples of endocytosis because you're bringing things within the cell. And then exocytosis is how, for example, a cell would release a hormone, right? It's going to release something out of the cell. Okay. Okay. So I find that this is rather straightforward material. You'll have no problems, I'm sure, talking yourself through this in that presentation. At the very, very end of this, I also give you a couple diseases about why we care about these different organelles. And at the very end of this, I have another review slide, and that's what I just want to go to right now. And that is right here at the very end of this, quote, first half of chapter 3. And it's another way of just thinking about the organelles. And I'm okay with your thinking about this as long as you understand the functions of these different organelles. This is a fine place to kind of look at it. If this is new information for you, then this is a great place to look. If this is old hat, if you've had the mitochondria now what seems like 30 times in your career, I get that. It is some information that is covered in a lot of classes. So if you just think of the mitochondria as being the powerhouse of the cell, right? That's where the ATP are made. If you think of the lysosome as being an area of the cell where you're cleaning up, a lysosome, they break things down. The lysosome breaks down dead and dying cells and organelles. If you think of the cell membrane as being the security gate, you can kind of think of this as like being a business, right? A small business. And so you've got a security gate outside the doors. You've got the cleaning crew coming in. You've got the powerhouse. You've got the generator, the mitochondria. You've got the distribution center, the mailroom, and that's the Golgi apparatus, the Golgi complex. You've got the proteins being made on the ribosome. So if this is like a factory where you're making something, the ribosomes and the rough ER would be the workbench and the assembly line for those proteins to be made. And then the control center, right, is part of the nucleus. And then, of course, the building is made up of some sort of structure that's the cytoskeleton of the cell. So this is a fine place to look for review. So chapter 3 is really 2 parts. The first part is organelles. I've been through that with you. And the second half is where I'm going to slow down now and talk to you for the first time about this material. So, questions? Anything at all before you review this on your own that you're concerned about? Has everyone here had organelles before? For the most part, you're just going to dust it off and review it. You'll be fine. Okay. Any questions, let me know. Now, let me start talking to you a little more completely as we get into the second half of chapter 3. And this slide, actually, I had 3 slides back, right? It's just a review. So the end of the first half of chapter 3 and the beginning of the second half of chapter 3 has this same slide. And what you're looking at here is DNA. DNA is the double helical molecule on the left. And we've seen a little bit about DNA. So tell me, what are we looking at here? What's the backbone of DNA made up of? Right, the staircase, right? This is the handle, right? The double handle or the railings of the staircase. And what's making that thing up? Sugars and phosphate, sugar, phosphate, sugar, phosphate. Phosphate is negatively charged. So I'm telling you that. So phosphate is negatively charged. So that means that DNA overall is very negative. Okay? Very negative molecule. And what you're seeing is that DNA is wrapped up very, very tightly around proteins to make chromosomes. So what a chromosome is, in your nucleus, you have 46 chromosomes. 23 of those came from your dad, 23 of those came from your mom. They match up. That makes you a diploid organism with 46 chromosomes. So what a chromosome is then is a very, very long molecule of DNA wrapped around a series of proteins. Those proteins are shown in this cartoon as little octets, little groups of proteins, and those proteins are called histones. Histones. Okay. Histones. So what charge do you think those little histone proteins have? Positive, right? So you got this negatively charged DNA molecule wrapped tightly around this little positively charged protein, histone. And this is tightly wrapped, and it wraps in multiple different ways until you get this densely packed chromosome. It sort of looks like a wound up telephone cord, doesn't it? Now, some of you don't know what that is, right? Anybody here never seen a telephone cord? Really? Okay. It won't be long, right? And some of your colleagues will have never seen a telephone cord. But it kind of looks like a wound up telephone cord, doesn't it? And remember that the chromosomes, we can only see them in the cell cycle under the microscope during mitosis. So when the cell is actively dividing, that's when you can see these wrapped up chromosomes. They wrap up very, very tightly when the cell is dividing. But during most of the cell cycle, these chromosomes are kind of loosely wrapped. And they're so loosely wrapped that they're not visible under the microscope. So that's what a chromosome looks like in a cartoon representation. Tightly wrapped DNA wrapped around little positively charged proteins. So that's the chromosome. It's the most highly organized level of genetic material in your cells. And again, your chromosomes, you have 46 long molecules of DNA in your cells wrapped around those proteins. Again, we only can see the chromosomes during mitosis, when the cell is actually dividing. So in lab last week, and some of the things you were kind of working through on your own, was the cell cycle. So this is good for me to review this. The cell cycle is, in fact, the cycle that every cell in your body is in the midst of doing. Every organism has a life cycle. Every cell has its own cell cycle. And cells must go through this entire cell cycle to replace themselves. So at the end of the cell cycle, the cell divides and undergoes mitosis. And then those two cells go through their cell cycle, and then they divide to create more cells. So the cell cycle is very, very important for normal development, for you from an embryo through development, and also every day. You are making millions of cells. You're making more red blood cells. You're making more gut epithelial cells. So every day you're making somewhere around, I don't know, a few billion cells a day. It's pretty remarkable how many cells your body's making every day. When cells die, as part of their normal process, this is called apoptosis. That's a fun little word to say. It's a very important word for you to understand. If your body just kept making new cells and we weren't getting rid of old cells, we'd be, growing exponentially larger. So it's important that the body is getting rid of cells as fast as it's making them. Now, as a child, of course, you're growing more cells than you're losing. But as an adult, right, we're kind of stable in our size and the number of cells. And so our cells are constantly undergoing apoptosis. This is cellular death that's supposed to happen. It's the way it's supposed to be. Now, where this story, I think, is really important, this idea of the cell cycle, is in early development. I'm going to try to prove to you why we care about mitosis, because I know it seems kind of academic, and do we really need to look at this? But this is female reproductive system, and in this cartoon, fertilization has occurred. So what we have, let me just give you your bearings, and this will be our last lecture together this semester where we talk about reproductive systems. So you'll see this all again in lab and lecture. But here we have the vagina, the cervix in this area, the uterus. Off from the uterus are two fallopian tubes, so we're looking at one fallopian tube. The fallopian tube leads up to the ovary. And from the ovary, an egg will be released approximately every other month. So in this situation, the egg has been released, and the egg is released into this top part of the fallopian tube. And this is where fertilization most normally occurs. It's at the very top of the fallopian tube. And the reason is, sperm, while viable for about 72 hours, eggs are only good for about 24 hours. So if more than 24 hours have passed from the time the egg has been released, it's no longer good for fertilization. So that means that at the time that the egg is released into the fallopian tube, either sperm were already there, or sperm arrived shortly thereafter. Okay? And fertilization happens. So the egg and the sperm meet, and that's what we're seeing in this cartoon. You're seeing an egg up here, and many sperm are trying to get her attention, right? So you got all these sperm coming in. One wins the prize, and that one sperm now is going to be part of the fertilization event. That has now formed the zygote, right? The zygote, zyg means the union of. The zygote is the union of the egg and the sperm. We have this first cell, and this first cell is now going to divide, right? So that zygote's undergoing cleavage. It's going to undergo mitosis. 2, 4, 8, 16 cells. Those cells that zygote is dividing, that embryo is dividing. And by the time the embryo reaches the fallopian tube, it takes about 5 or 6 days for this journey. And during that 5 or 6 day journey down the fallopian tube, the cells are dividing. The embryo has to be sufficiently developed in order to grab onto the uterus and hang out for the next 8.5, 9 months. So it's very important that the mitosis occurs very quickly. By the time we get through those 5 or 6 days, you must be at a solid ball of cells called a morula. We'll get more to this in lecture in about a week or so. But in addition, you've got to get to the blastocyst stage. And the blastocyst stage is basically the embryo ready to grab on and reach into and grab the uterus wall. Okay. So clearly, mitosis is important for normal development. It has to be going really, really fast, and everything has to be working well. Now, another place where people study mitosis, not only in early development, but also in cancer, right? Because what is cancer? Cells that are growing out of control really, really fast. So they're completing their cell cycle, and they're growing out of control. So half of the work, if not more, of the work that's being done at the Van Andel Institute, right, research, scientific cancer research, deals with regulating the cell cycle, deals with regulating mitosis. How can we stop cancer cells from dividing? How can we attack them? How can we stop those cells from dividing while not telling the rest of the body to stop dividing those cells? So because we know that this is a 6-day journey down the fallopian tube, there's no doubt that fertilization happens a lot more often than implantation. Right? So egg and sperm are going to meet more often than implantation. In fact, it's also possible, we'll talk more about this at the end of the course, but the egg, when it's released, can not be sucked up into the fallopian tube. It can hang out in the abdominal pelvic cavity of the woman. And sperm can certainly just keep on going into the abdominal pelvic cavity as well. So it's possible for fertilization to happen outside of the fallopian tube. And that would be one example of an ectopic pregnancy. Now, the woman would never know fertilization never occurred because that embryo has no place to attach. And it would simply die. It would undergo apoptosis, if you will, and would not have any chance of surviving. Okay, so brings us back to mitosis. Really, really quick. Mitosis is derived from the Greek word meaning thread. So when scientists first looked under the microscope and they looked at cells, they saw that some cells had these thread-like structures. We now know that those thread-like structures are the chromosomes, right? So thread, mitosis. So it's a condition where there are threads, if you will. They didn't know what those things were, right? But now we know what they are. Now, the cell cycle, 3 phases, G1, S, G2, and mitosis itself. And mitosis is simply the time when the threads appear during the time of cell division. G1 and G2 are just normal growth phases. They're just when the cell does what the cell does, growth or gap. S phase, what does the S stand for? Synthesis. What's being synthesized? DNA. So that's where the chromosomes are being duplicated and where the cell's also making sure it has plenty of mitochondria, has plenty of Golgi, it has plenty of all the other things, because soon, very soon, what's going to happen? The cell's going to divide. And when it divides, it needs to make sure it has 2 of everything, right? It has to be able to split down the middle. So that's happening during the S phase. And then when the cell gets to mitosis, this is a relatively short period of time, in fact, very, very quick, highly orchestrated, where the cell divides. So you've kind of broken this down a little bit in lab studies, so you know that there's that prophase and anaphase, and metaphase, anaphase, telophase stages. So I've got those on here again. Prophase, P for puffy, the cell kind of gets puffy. The chromosomes line up in the middle for metaphase. They're being pulled apart for anaphase. And finally, T for 2, we're getting 2 cells as telophase finishes. The other way of thinking about that is that telo means end, right? So telo means end. And so telophase is the end phase, the last thing. At the end of all this thing, we've got cytokinesis. So here is the cell cycle, a cartoon of the cell cycle, including mitosis. So the problem with this picture is that it makes it look like mitosis takes half of the cell cycle. Time-wise, this is not true. Time-wise, mitosis is a very, very thin little slice, but this at least shows all the stages, including the stages of mitosis. So I don't want you to be drawn off by that. So probably the best place to start a conversation like this is just to look at where one cell becomes two. So at the moment of cytokinesis, you now have two cells. We're going to follow this cell right here. So this cell is now in the G1, growth 1. Some books call it the gap 1 phase. And this is where the cell does what cells do, right? It's growing, it's responding to its environment, it's getting in nutrients, it's doing all the things we talked about being the characteristics of life. Notice when I look at this cell, I see that the two centrioles, right, are kind of on one side. They're not spreading out. And I can see a nucleus, I can see a nucleolus, but I don't see distinct chromosomes. So that would be typical of a cell in the cell cycle, somewhere in G1. Then in S phase, we can't see anything happening here, okay? But just know that during the S phase, that's where all the chromosomes are being duplicated. So now, a cell coming out of S phase, going into G2, has how many chromosomes? A normal cell has 46. After S phase, we've now duplicated everything. There literally are now 92, sorry, get my math right, 92 chromosomes. Right? There are 92 chromosomes in that cell. Because of what happens when the cell divides, we split those 92 back into 246s. And so we have to have everything duplicated during this G2 phase. And then we go into mitosis, the 4 stages of which are prophase. Remind me, folks, what's going on in prophase? 3 things.
Chromosomes are visible.
Look at the pictures, right? The chromosomes, we start seeing those chromosomes. The spindles have gone to their, well, the centrioles have gone to the north and south pole of the cell, and we can't see it here, but they're going to start making those mitotic spindles. And thirdly, the nucleus has dissolved, broken down, gone away. Okay? From prophase, we go into metaphase. Very distinctive. The chromosomes line up along the border, along the center, equator. Then during anaphase, then those microtubules are going to start pulling, and those duplicated chromosomes are going to be split, right? So that now each cell gets one copy of what was made back in the S phase. Then we get to telophase, T for 2. You see the cleavage. And finally, the moment of separation is called cytokinesis. Around and around and around it goes. Quick pictures of this. Again, this is just blown up pictures of the same thing. So this is interphase. Again, what do we not see? Yeah, I don't see any chromosomes in the nucleus, and the centrioles are hanging out together. Then in this phase, the nucleus is gone, right? The nucleus would be around here. So I'm starting to see the chromosomes, and the centrioles are on either side. That is prophase, as it's marked on the bottom. Just so you can see this really quick. Metaphase, lined up in the center. Anaphase, being pulled apart. And then telophase, cleavage, separating. You should be able to recognize these in a cartoon. You should also be able to recognize these from the pictures, the actual micrographs down below. Now, there's another type of cell division, one that I will not be spending a lot of time on here. We'll come back a little bit to it when we get to the reproductive systems at the end. Mitosis is the way by which almost every cell in your body has been formed and continues to make more. There is, however, another type of cell division called meiosis or meiosis, and this is a type of cell division occurring only for the production of your sex cells or your gametes. So they're the only cells in your body produced by that are involved with meiosis. Now, meiosis starts off with a regular old cell. And that regular old cell is a diploid cell. Dip means 2, right? So we got 2 of all the chromosomes, diploid. And in this process of meiosis, at the end, we're going to end up having haploid cells. Haploid cells, haplo means to have cut in half. Okay, so we're going to have half as many, or think of H for half. So we're going to cut the number of chromosomes in half. That means that each egg or each sperm is going to have how many chromosomes? 23, right? So a normal cell, diploid, has 46. and the egg or sperm are going to have 23. So its only place in the body where this is taking place is in the formation of your gametes. The gametes are a collective name for egg and sperm. And where are the gametes made? What organs, what tissues make the gametes? Yeah, the gonads, right? So that's a collective word for testes and ovaries. Now, another way of referring to this, it's not exactly the same, but meiosis is very important in the overall process of gametogenesis. Look at this term for me. Gameto, gametes, genesis, in the beginning, the formation of. So we've got the formation of the gametes, and to make your gametes, you have to use this process of meiosis. Okay, so why do we care? I tried to convince you a few moments ago that we care about mitosis because to study cancer and to understand early development, mitosis is an important indicator of all that going well. In meiosis, here's a reason why you care. This young child has Down syndrome, trisomy 21. Okay. And this is an example of meiosis gone wrong. So in development, in the production of this child's father's sperm or mother's egg, meiosis didn't go well. And when the chromosomes were dividing, rather than dividing equally, in the case of this child, there was an extra 21st chromosome. So if you look in this picture, again, here's our normal cell, and it's going to divide. And when it divides, it's only going to have half as many chromosomes, so we call it haploid. Typically, all the chromosomes would equally divide and go to one side or the other. In this case, look what happened. These two chromosomes, rather than separating, went together. So that means that this gamete, egg or sperm, carries, in this case, an extra 21st chromosome. It could have been a 23rd, a 24th, 23rd. It could have been any of the chromosomes, right? But this is the 21st chromosome that creates this problem with Down syndrome. Now, the human, we can handle a few examples of extra chromosomal information. Now, life for a person with Down syndrome is not normal, but it is viable, right? There's life. So it is possible for a person to live with an extra 21st chromosome. It's not possible for a person to live with an extra number 1 chromosome, or a number 2 chromosome, or a number 3 chromosome. And the reason is the chromosomes are numbered in their size. The first chromosome is huge. The 21st chromosome is much smaller. And so it's possible for life to go on, kind of normal, for a little bit of extra genetic information. And when that extra genetic information from the 21st chromosome is a part of your genome, then you have things like heart disease, funny palm crease, the facies of Down syndrome, usually some significant mental challenges as well. The life cannot go on without chromosomes. So if instead of this egg or sperm being used, if this one had been used to create this embryo, life would not go on. Okay? So there has to be 2 of every chromosome. The only exception to that is the X, right? Men don't need 2 X's, and women, you don't need a Y at all. That's the only exception. But all the other chromosomes, we all require 2 of every chromosome to survive. So that's why we care about Down syndrome. It's one of many examples where meiosis went wrong. Okay, just as a, why do we care? I've also mentioned the word apoptosis. Apoptosis is where cells die because they're supposed to, right? It's normal cellular death. So here is a human embryo going from week 5 to week 10. So this little guy right here, right, this embryo, about the size of a quarter. Okay, that's week 5, and as you progress over, this is week 10. What you'll notice, what I want to draw your attention to are the hands and the feet. At week 5, the entire arm is like this little short paddle. There's really no, it's like a little oar. There's really no structure to it. There's no fingers yet. And over the next 5 weeks, you start to see the arm extend. You can see the arm extending. And by the time we get to week 10, that could be your hand, right? It doesn't look any different than your hand. Little tiny hand, perfectly formed. And on the bottom is a person's, or is an embryo's 10-week-old feet held between adult-sized thumb and finger. Right, so this is perfectly little formed toes, perfectly little formed fingers. That's week 10. How did that happen? Well, it turns out that that paddle, that solid structure, apoptosis had to occur. So there was actual cell death that occurred down the center to create the palm, okay, to create the fingers. Now, what if that apoptosis did not occur?
Webbing.
Webbing, right? So some people are born with webbed fingers. And they're born with what's called syndactyly, where their fingers are webbed together, or their toes. Or they may have extra fingers, and that, again, is a defect in this process. So somewhere between weeks 5 and 10, this process is occurring, and apoptosis was critical. Now, another way of thinking about it, what if right now every one of my cells in my arm decided to divide? My arm would be twice as long, or twice as big around, right? I could be scratching my toe right now just doing this. So the point is, our cells know when to divide. They know when not to divide. It's pretty amazing, right? Every cell in your body knows when to divide. It has the right signals. Bone cells grow at a different rate than skin cells, that grow at a different rate than liver cells. And neurons never divide. So once you have a nerve cell, a neuron in place, that's it. They don't divide. And the reason neurons can't divide is they don't have centrioles. So if you were a cell without a centriole, what can't you do? You can't spit out mitotic spindles, and you can't do mitosis. So neurons do not have centrioles, so they cannot divide. So once you have a neuron in place, that's it. Right? And so as you lose neurons with age and other activities, then you never get those neurons back. Okay, so that's apoptosis. And I've already kind of mentioned that we care about mitosis not only for early development, but for tumors. So what is cancer and what are tumors? Now, there's all sorts of cancers. I'm going to talk just about solid tumors here for just a moment. So a solid tumor is a ball of cells. It's out of control. The cell, for whatever reason, is not dying, and more than likely, it's just dividing out of control. There's a problem in control of the cell cycle. And this whole little ball of cells becomes what's called a tumor or a neoplasm, a new growth. Okay, neoplasm, a new growth. So there's two basic categories of cancer. Now, it's far more complex than this, but two basic categories of tumors, benign and malignant. So benign neoplasm is simply it's cancer, but it's typically contained, and it's going to grow rather slowly, and it's more than likely not going to kill you. These cells tend to behave, they grow more slowly, and they don't spread as quickly, if at all. So a couple terms that are in here. These are still cancer cells, and cancer cells, by definition, de-differentiate. Let me try to describe that word to you. You have in your body about 220 different kinds of cells. Every one of those cells came from where? At one point, you were a zygote, right? One cell. That one cell became 2, became 4, became 8. Somewhere along the way, those cells in that ball became different from each other. There were instructions given, and that process is called differentiation. So along the way, liver cells, brain cells, stomach cells, you name it, are becoming different from one another. That's the process of differentiation. Cancer cells de-differentiate. Basically, they become more childlike. They become more adolescent. And what did we just see happens in a growing embryo? Early cells grow fast, right? The 2, 4, 8, 16 cells that happen during the fallopian tube are rapid cell growth. So these cells become de-differentiated. They go backwards. We used to think, and this is the last 5 or 10 years, we used to think that once a cell received the instructions to become a muscle cell, that was it. A muscle cell was going to be a muscle cell, a liver cell was going to be a liver cell, a kidney cell was going to be a kidney cell. In the last 5 or so, 10 years, they've now been able to take muscle cells in the laboratory and add the right chemicals at the right time, de-differentiate those cells to a more simplistic cell, and then drive forward to make skin. So we've been playing around with skin cells and muscle cells, and we're beginning to understand in the laboratory, trying to understand how the body does this and trying to reproduce this. So we can now take cells in culture and play around with them a little bit. So cancer cells are de-differentiated. They're more childlike. They're growing faster. They also tend to get their own vascular supply. Tumors, to survive, right? You get this massive ball of cells. Every cell needs to survive with blood and oxygen and nutrients. And so tumors tend to get their own blood supply. They become very vascular. And one of the major targets of chemotherapy agents is to block the ability of the tumor to get its own blood supply. So we'll talk a little bit about that. Tumors are, these again are normally not lethal. They're only going to be lethal if they're in a non-surgical place and they're slowly growing, maybe deep in the brain, or maybe they're growing up against an important structure and they can't get to it. It's too embedded in the important structure. But typically benign tumors are not going to kill you. Now what does Benny mean? The prefix. Good, right? This is good news. I mean, if you're going to get tumor, this is the one you want, right? Benign. Good. Good stuff. Now, also, cancer is, another group of them is malignant neoplasms. Now, the difference here is that if it's a malignant tumor, oh, I should go back. I said it, but I didn't focus on it. Benign tumors typically are confined within a connective tissue capsule. In other words, they're confined. You can go in and scoop it out, and you can get the entire tumor. You can get the whole thing, surgically remove it, or zap it with radiation, but usually you can get the whole thing out. Versus a metastatic cancer, and what happens here is that now these tumors are unencapsulated. They don't stay within a certain area. They're still differentiated. They still get their own vascular supply. They still are growing very rapidly, but here's the kicker, they spread, right? They metastasize, which means they go beyond their normal places. So these cells will break off from the tumor and spread and go to places where they shouldn't be and set up a secondary cancer. So this is why some lung cancer spread to the brain, or why some brain cancer spread to the bone, right? It's metastasis, and this is ugly. So this is when you have to start doing chemotherapy. If it's a benign tumor, or regardless, what if a person sees a little tumor in their chest? Let's talk about breast cancer for a second. And there's a breast tumor seen in a mammogram. What can they do? They can go in and they can do a biopsy. They can remove it. And typically when they're removing that tumor, if it's small enough, they may do some radiation and shrink it down, then take it. They're also going to check the lymph nodes out nearby. Now those lymph nodes are places where cancer cells like to go. We'll talk about lymph nodes later in the course. If those cancer cells are not in the lymph node, and they do enough of these to statistically convince themselves, if there's no evidence of cancer in the lymph nodes, then what do they assume? The cancer has not spread. It is likely contained. And when they do the biopsy, they can see if the edges of the tumor are smooth and they've got the whole thing. So they can look at that under the microscope and look at testing from the lymph nodes. If instead they see evidence of either during the biopsy or cells in the lymph nodes, now we know the tumor has metastasized. It's probably gone far beyond just the lymph nodes of the breast. It's anywhere. So now we got to go chasing after that tumor, right, with chemo agents. And what's the purpose of a chemo agent? To kill, stop the rapidly growing cells. But what's the problem with chemo agents?
Breaks on other things also.
There's a lot of side effects, right? What are those side effects?
Sick.
Sick.
Hair loss.
Gut, right? Diarrhea, vomiting, a lot of discomfort in the GI. What else?
Weight loss.
Weight loss, because they're not eating well. Right, so you're killing all the cells, but what are the other symptoms, the other side
Hair
effects?
loss.
Hair loss. Things don't taste right. Taste buds get all messed up. Hair loss. Feeling really anemic, right? Really lousy, low energy. What are all those things connected to? Hair loss, gut, diarrhea, and vomiting, taste. What's all that got to do with each other? You're killing rapidly growing cells. Hair is rapidly growing. So you're killing those cells off. The gut lining, the epithelial cells. So you're damaging the epithelial cells. You're not going to eat well. You get upset stomach, a lot of diarrhea, vomiting. Taste, nothing tastes right because you're killing off the taste bud cells. And you're killing off your red cells. So you don't have enough oxygen. You're anemic. You're killing off your white cells. That's part of your immune system as you become immunocompromised. And you're knocking off your platelets, which are involved with blood clotting. So all of those symptoms, right, of chemo, don't feel well, low energy, hair loss, vomiting, all related to because these tumors, or sorry, these medications are chasing after those rapidly growing cells. So it makes sense to us why those symptoms occur. Okay, so just a note or two about cancer. So again, the cancer cell has lost its control of its cell cycle. It's growing out of control. It's also true that cancer cells usually have most cells have contact inhibition. If I took some skin cells out of my arm and put them in a petri dish, gave them the right conditions to grow, they would grow to the edge of the dish and stop. Or they would grow until they touched another cell, and then they would stop. So most cells know where to grow, and they know when to stop growing. Cancer cells have lost control of this. They've lost it, so they start growing where they shouldn't go. They roll on top of each other. They make these solid balls. So they overgrow one another, and again, they also get their own blood supply. And just like the benign tumors, they're dedifferentiated, and they get their own blood supply. One way of saying that is angiogenesis. So what does angio mean? We know this one. Vessels, right? In Genesis, in the beginning, the formation of. So we've got the formation of blood vessels. Angiogenesis, those tumors need that blood supply. And then here's that big one, right? These cells, these metastatic cells are going to be invasive. They're going to break off and travel through the bloodstream and through the lymphatic vessels. And that term is metastasize. So malignant tumors tend to metastasize. They spread. So hopefully you care a little bit more now about mitosis, right? Because most science research, most cancer research is dealing somehow with mitosis and understanding all the signals that are trying to control cell cycle. Now, certainly not on your radar today, but on exam number 2, there will be a short writing assignment. That short writing assignment will be as follows. I will give you 11 terms, and here they are, okay? So, not lecture quiz 1, but this will be lecture exam 2. Okay, I need to update that. Lecture exam 2. And here are the 11 terms that you need to learn. Some of these we've already talked about. Some of these I'll be talking about over the next few minutes. Transcription, nucleus, DNA, translation, proteome, cytoplasm, ribosomes, proteins, rough ER, genetic code in mRNA. And with those 11 words, you must use 10 of those 11 words to write 3 sentences. It's really not that tough, but what it proves to me is if you can write 3 cohesive sentences using these terms, you understand the relatedness of these terms one to another. So, right now, which words have we already talked about? From the organelles?
DNA.
Yeah, DNA is in the nucleus, right? Ribosomes make proteins on the rough ER. So some of those terms we've already talked about, and then I'll be talking about transcription and translation today, proteome. I will give you those words. I'll talk to you about it again. It's not something you're going to worry about at all in the next couple of days. Okay. That's exam number 2. Let me just pause right here. Anything at all about cancer, about cell cycle, about meiosis, Down syndrome, anything like that that I've discussed, apoptosis? Anything at all? Now, you will not find that presentation described that way in your book, okay? So just know that's kind of more of a background, why we care, kind of introduction to all this. You will find the rest of this, though, laid out for you. So we're going to go into the cell now. We're going to go back into the nucleus, and we're going to see DNA there. So here we are talking about DNA, and here's a double-stranded DNA molecule, and we've already said that the DNA is wrapped around these proteins called histones to make a chromosome. This is a new word for us, chromatin. Chromatin is the combination of the DNA with the proteins. So you would say that your chromosomes are composed of chromatin. Chromatin is composed of the DNA and the histone protein. So it's kind of just a building of these terms. So DNA plus proteins makes chromatin. Chromatin is the building block of your chromosomes, and those chromosomes are found in the nucleus of the cell. No biology textbook is complete without a picture of these guys, right? This is Watson and Crick. We've all seen this picture before, probably. I always kind of am amazed. These guys won the Nobel Prize without really discovering anything. I mean, what they did is they played with some Tinker Toys, and they were the first people to figure out the structure of DNA. Pretty amazing. And yet they did no real experiments. They really ripped off everybody else's work. So what they were doing is just kind of watching the other works of other scientists. I'll mention a couple of them today. And from that, built this model, this double-stranded model of DNA. Now, what's really remarkable, though, about their paper from 1953 is that they said, if this is the structure of DNA, then this must be how the whole thing works. And they did hypothesize quite accurately how transcription and translation would occur from this structure. Before that, no one really understood. Was it DNA or was it protein that was the most important molecule? Which molecule was responsible for heredity? That was debated even long after Watson and Crick's discovery, and they described everything. It was not, everyone was not convinced. Some people thought that proteins were still the most important molecule in your cells. So the woman that they really ripped off from is here. This is Rosalind Franklin. Now, she had done some work. This is a X-ray diffraction image. Not that I understand this, nor do you have to. But basically what she did is she took purified DNA. They knew about DNA, right? They knew about chromosomes. They didn't know everything about them, but they knew they could isolate DNA. So she isolated DNA, and then she shined X-rays through it. And when you shine X-rays through any molecule, it'll spread and make a pattern. So this pattern, this X-ray diffraction pattern from her, told her and Watson and Crick that DNA was double-stranded and that it was of a set diameter. Okay, so it was a certain size based upon this, and it was a double helix. And that's what she contributed to understanding the model when they put it together. Now, she passed away very young, probably because she shot around X-rays all day, right? She died of cancer very, very young from that X-ray exposure. Back in those days, you know, Madame Curie and all those folks played around with radiation and radioactivity, didn't know what they were doing. They didn't realize the danger of it. They never give the Nobel Prize to someone who's already passed away. So in '62, when they awarded Watson and Crick the Nobel Prize, they gave it to her co-worker, Maurice Wilkins, but they did not give it to her because they just don't ever give it out to a person who's already passed away. So unfortunately, she was not included in that Nobel Prize. And it's still that same way. So when I was at the University of Florida in the college, we would have these little bets on who was going to get the next Nobel Prize. And it was sort of a combination of great discovery and old age. Because, you know, they'll never give it to anyone who's already passed away, and they usually don't give it to really young people unless it's a remarkable discovery. Okay, so if you get the Nobel Prize, maybe they think you're getting old. I don't know. So we've already talked about DNA and RNA, right? The major difference is, just to recap that, a little bit more detail here than what you need to know for the exam. So as we've already said, there is a sugar-phosphate backbone in nucleic acids. In RNA, that sugar is ribose. In DNA, that sugar is deoxyribose. That's where we get the name, deoxyribonucleic acid, DNA. So what you have is, again, in RNA, you have a sugar called ribose, and in DNA, you have a different sugar called deoxyribose. It's a very small difference. I'll tell you why that's important in a moment. Then, as we've already said, we know this one, RNA has A, C, G, and U, whereas DNA has A, C, G, and T. And RNA usually hangs around in the cell as a single-stranded, a single linear molecule, whereas DNA is most often found double-stranded, wound around itself. Now, here's why RNA is single-stranded most of the time. This ribose versus deoxyribose sugar. This is a ribose. You don't have to memorize that, but that's a ribose, and this is a deoxyribose. The only difference is this oxygen group. Okay, I'm not going to quiz you on this, but I want you to appreciate this. The only difference is that oxygen group. That oxygen is kind of a big, clunky molecule. And as a result, it doesn't allow RNA to hang out in double-stranded very well. So the major difference that keeps DNA and RNA one double-stranded, one single-stranded, has all to do with this oxygen. So we've got a ribose versus a deoxyribose. Here's that phosphate group. It's on both. So on both of these, we have the phosphate group. I already told you that phosphate groups are negatively charged. So you've got this sugar phosphate backbone, and DNA and RNA both, by their nature, are negatively charged molecules. Again, I'm not going to ask you to recognize or draw out these structures. This is a cartoon representation of the double helix. Again, the backbone or the staircase, right, the handrails of the staircase, sugar phosphate, sugar phosphate. If this is DNA, that sugar is what sugar? Deoxyribose. And then the steps of this staircase are the bases. And the 4 flavors are A, C, T, and G. So what we see here is that A and T always line up together, and C and G always line up together. Now this was another thing that Watson and Crick figured out when they were making their Tinker Toy model of DNA. And they ripped this information off from somebody else as well. But someone else had described that it seemed that the concentration or the percentage of C's and G's in a cell was always about the same. That the concentration or the number of A's and T's were always about the same. Also, what did Rosalind tell us? That from her DNA diffraction data, that DNA was of a set diameter. If you look at that diameter and you look at these molecules, what you're going to find is that A's and G's are these double molecule, these big molecules, right? A's and G's are this double molecule, and those are called I'll get to the base names in a minute. And then the C's and T's are single. So what they figured out is that one of these big ones is always matched up with one of the little ones. One big one's always matched up with one of the little ones. So what do we see? A's and T's always match up, don't they? And C's and G's always match up. If 2 of the little ones matched up, the molecule would be too narrow. If 2 of the bigger double molecules matched up, the molecule would be too wide. So it had to be that an A went with a T and a C went with a G. And that's what I'm showing down here. We call these complementary bases. It's hard for me to write this, but A equals T, the equal sign, there's 2 hydrogen bonds. Remember hydrogen bonds? Those weak bonds between hydrogens and other molecules. And C's always go with G, but they're connected by 3 hydrogen bonds. So you got this equal sign with a line underneath it. So you have 3. So basically then DNA molecule is like a zipper, right? You could really kind of pull the 2 parts. Those hydrogen bonds are not that strong. They're just strong enough to hold the darn thing together, but you would, without much trouble, be able to separate these strands. And that's one thing that the cell does do when it's making more DNA. It does have to separate rather easily these 2 strands. Now, let's look at another breakdown of these bases. And again, what we see here is the A and the G have like 2 ring structures side by side. Those molecules are called the purines. Okay, that double structure is called the purine. And the green ones, the single molecules, are the pyrimidines. So the easy way to remember this, I think, is that to make the pyramids, you have to cut the stones, right? So CUT, pyrimidines, pyramid, close enough to remember that. And then I've heard other people have learned that what is the chemical symbol for gold? What is it? AU and AG, right? Those are the metals like pure gold, pure silver, pure, right? So the purines A and G. Okay, so either way, I always think the pyrimidines cut, right? CUT. Well, that just helps you remember. So then what does that mean? That in the DNA molecule, you have one purine matching up with one pyrimidine, right? One purine matches up with one pyrimidine. So we're back to the cell cycle again. And now I'm focusing on the S phase. You've already told me correctly that the S phase is the time when the DNA is being duplicated. The fancy word for that is replicated. If you make a replica of something, you're making an exact copy. So that's the idea, that in the S phase, the cell is making an exact replica through the process of replication of the DNA. So how this works is really quite ingenious. And again, this is what Watson and Crick described in their paper. So I always give them a hard time. But they did an amazing thing. but I still think they played with models. They just got it right. Okay. Other people tried. They got it right. So basically what we have here is that this is one strand of the DNA, and this is the other strand of the DNA. And how do these things go together? Called complementary basing. So pairs. So the A will always match up with, the A will always match up with T, and the C will always match up with G. What's missing here is that the C and G are combined by 3 hydrogen bonds, and the A's and T's are bound by 2 hydrogen bonds. That's just not shown here. So that's basically how the cell makes more DNA. It takes one strand, right? Takes half of it, opens it up, and makes a duplication or replication of each piece. Now, we're not going to go into the details of that. If you take a bio course, cell biology course, you'll spend hours and hours and hours learning the hideous details about replication. But what I want you to realize here is that replication is really, really fast. In the cell cycle, in order for us to copy all the DNA, we've got a lot of DNA to copy in our cells. And so this has to be a very, very fast process. In bacteria, E. coli specifically, where we learned most of, and I say we collectively, the scientific community, learned most about replication from bacteria because they grow so fast. So we learned that E. coli can copy its DNA in less than an hour. And to do so, it has to copy about 500 nucleotides, ACTs and Gs, per second. That's a pretty amazing process of copying. Now, in you and me, our machinery, we have about 6 billion base pairs, 6 billion ACTs and Gs to copy, and we have to do this in about 8 hours. The fastest growing cells in our body are like epithelial cells, your gut lining cells. And they can replicate and they can divide every 8 hours. And most of the dust in your house is you, right? Your skin cells. So those skin cells, that's what's dividing really, really fast. In order for our body to divide in 8 hours, those cells need to copy about 50 nucleotides per second. That's still a pretty amazing rate. The problem is, going so fast, errors are made. Right? So we've got an issue here, timing and quality control, just like Toyota does. Right? We've got to make them fast, but we have to make sure the quality is good. That's really what the cell is dealing with. So when the cell is dividing and when the cell is undergoing replication, if there's a change in the DNA through mistake, we call that a mutation. So muta means change. So a mutation is simply a change in the sequence of ACTs and Gs. Now, your cells make about a mistake every 1 in 100,000. It's pretty good. Right? Toyota would be really excited about only making one lemon car, 1 in every 100,000. So it's a pretty good rate, but gee, I just told you there's 6 billion letters in every one of our cells. So that means that our cells are making quite a few errors even every time they copy. Right? There are lots and lots of changes by errors being made in our DNA. Now, if those changes are in a somatic cell, now the word soma, the prefix soma, means body. So these are all your body cells, your somatic cells, all the cells of your body except for your gametes and gonads. Okay? If you have an error, for example, if you have a mutation in your skin cell, well, okay, you may get skin cancer in 20 years, but it's not going to be passed on to your kids. Right? So you can sit in the tanning booth all day, and your kids are not going to inherit that cancer. Right? You're only going to pass on your mutations if those mutations occur in your germline, which is a fancy way of saying the cells that will become your egg or sperm. So you can sit on the tanning bed, you can have, you can bathe yourself in chemicals that cause mutations. That's not going to get passed on to your kids. It's not heritable unless it is passed on through your germline. Okay? Which is why when you go and get an X-ray, right, if they can, they'll put a lead shield over your gonads, right, if they can, because we want to protect those germline cells from additional mutations. So what we see here is just a cartoon representation of the starting DNA sequence. We've got C's and G's matched up with A's and T's. All is good. Life is good. We undergo replication. In this rapid in this rapid copying process, something went wrong. And what you see here is that a G got matched up with a T. Now that's not right. Okay? And the cell will notice that error and will try to correct it. The problem is the cell doesn't know, was the G correct or was the T correct? Okay, it can't always tell which one was put in there correctly. So it may guess, and it may guess incorrectly. In this case, it guessed that the T was the original letter, so it fixed the problem and put an A in there. A and T are matched up properly, but what has happened? We've had a mutation, haven't we? There's been a change in the DNA. You've gone from what should have been a GC to an AT pairing. If that does not get caught or changed, and that change is a permanent record in the DNA of that particular cell, then a mutation has occurred. And every cell that that cell makes from that point on will have that same mutation in it. So if I get a mutation in my skin cell, right, in the skin cell on the back of my hand, that's fine. But every cell that is created from that cell, right, as that cell divides and as those cells divide, all those cells will also have that same mutation. Okay, so we have to think about it that way. It's not just affecting one cell, but it's affecting every cell that is made from that cell forever and ever. Now, mutations are going to come in two basic types. Again, you could have an entire class on mutations. But basic mutations. Number one, chromosomal mutations. These are going to be errors in meiosis, for example, Down syndrome. So this is where the chromosomes don't behave, they don't divide properly. That's not what I'm talking about here. The kind of mutation I want to talk about here are point mutations, where one little point, one little A, C, T, or G is switched or changed, or maybe a few of them. Now, these point mutations are usually the results of problems in replication. So I told you there's about, it's ripping along at about 50 letters per second. One out of every 100,000, there's a mistake made. That's what I'm talking about. Those are the replication errors. And in addition to the errors in our own copying mechanisms, there's also environmental issues. Radiation, X-rays, chemicals, smoke, those things that are coming in that environmentally are causing damage as well. So we have all sorts of things bombarding us. If you fly in a plane, you're being bombarded by all sorts of rays in the sky. In this building, we're in the basement. We're under the dirt with a lot of cement around us. We're probably getting bombarded right now with some more radiation than a person on a higher level. Any kind of carcinogens or any kind of chemicals that you consume or breathe in that are known to be over time in a high enough concentration, carcinogens, this is also a problem for making these point mutations. Ultraviolet light, okay, all those things. UV light going out in the sun, spending too much time at the beach in the summertime, is all of those things can cause these point mutations. Now, the good news is that even though we're getting these constant mutations or potential mutations in ourselves, our body has a lot of ways to fix it. So collectively, in all of the different repair mechanisms, there are over 130 enzymes and proteins involved with repairing your DNA. So our DNA is constantly being bombarded, it's constantly being mutated, but we have a whole army of mechanisms to fix it. So basically, that's the good news. Right? The reality is most of us carry between 5 and 10 disease genes, okay, within our genome. Now that's on average. Some of you may not have any, but most of us have 5 or 10 different genes in our body that we can pass on to our kids that can cause disease. And unfortunately, if a couple get together and they happen to carry some of the same genes, right, the same mutations, and their kids are at higher risk for some of those diseases unknowns to the person. So it's important that we understand these mutations, right, these mutations, and we'll talk about mutations again at the end. Here's a representation of cancer in men with age. So across the bottom we have age. On the side we've got the incidence per 100,000 of males for different cancer types. So, age 60, we start to see cancer rising, and by the time a person gets to 80, now it's not like everyone, but 350 out of 10,000 will be having prostate cancer. Okay, quite a few numbers of skin cancer. Lung cancer is not on this picture. Just to give you an idea, with time, what's happened? Not only has our body been around longer to accumulate more of these mutations, but we've been exposed to more chemicals and environmental issues throughout our life. And we know, for example, in colon cancer, it takes between 4 and 7 mutations for a cancer to be initiated. That's in the same cell. So think about your own body for a second. Think about the back of my hand. If I get a zap of UV radiation and I get one mutation in that cell, and the body doesn't fix it, and it stays a mutation, that's fine. That cell has one little mutation in it. If this were my colon, we know that the same cell has to have between 4 and 7 mutations. The same cell. But how does that happen? One cell gets zapped, right, mutation, and it makes new cells that have that same mutation in them. And then that daughter cell gets zapped. Now that daughter cell has 2 mutations, and every cell from it has 2 mutations already built in it. And then that one of those daughter cells gets a third zap. So it takes 4 to 7 of these zaps, so these changes in DNA of certain genes, in order for cancers to get started. Now, some people are born with these, right? They inherited it from their parents. They're already higher risk. We all know, unfortunately, some families have more history of cancer in them. Those family members are inheriting a higher risk of cancer than others. Others are born with a cleaner slate. Some are born with a dirtier slate, if you will, greater chances of mutations. And just because a person has cancer doesn't mean they have the genes, but maybe along the way they picked up these environmental signals disproportionate to other people. So every single second, your body has 25 million cells undergoing mitosis. What does that mean? You're making 25 million new cells every second. What does it also mean? Apoptosis, you're also losing about 25 million new cells, or old cells every second. So you've got this constant turning over of cells. That's an amazing rate, right? We got 6 billion base pairs per cell times 25 million per second. So our mutation's going to happen. Clearly, we're going to have some errors in this. So when there is a change in the DNA, the cell has 3 choices. Repair it. I told you there's 130 or so different ways to proteins and enzymes that are involved with repair. So if the wrong ACT and G is put in there, there's a way to fix it. Great. We repaired it. Number 2, we can apoptose. If that mutation is so deleterious that the cell can't survive, then the cell will just apoptose, right, and just die. So now the host, the organism, is safe, that one cell's gone. Or, number 3, live with it, right? That mutation is now a part of that cell and will be a part of that cell forever and ever, amen. So it's sort of like your relationships, like your family, right? Right? You either fix them, kill them, or live with them. It's the same thing, right? Same thing. Make sense? So now these changes in our DNA, if we don't fix them and the cell doesn't die, those DNA changes can over time lead to what evolution biology tells us is the basis of change, right? So over time, those mutations that are occurring randomly can lead to changes, most of which are negative, but some of which are thought to be advantageous. And so over time, this is the mechanism of evolution. Any thoughts in that conversation? I'm going to take about a 5-minute break here and give you a little break. But any thoughts about anything I've talked about so far, let me know. We'll come back in 5 minutes, and I'll talk about transcription and translation. Any thoughts, questions, concerns over anything I've talked about so far? Anything at all so far?
No.
Yes?
Chromatin is DNA plus protein?
Yeah, so the chromatin is the word given to the DNA wrapped around the proteins. So you would say the chromosomes are made of chromatin. Chromatin is the DNA in the protein. So it's just another layer of, they wouldn't be happy to not have an extra word, extra term. Yeah, question?
So the chromatin is just half?
Okay, chromatid. I didn't go there. There's a lot of chromatid words, right, in genetics and cell biology. Chromatid would be that DNA, when you see the X,
Yeah.
right? The X is actually a duplicated chromosome. Each part of that X is a chromatid. I'm not going there. Okay? Not part of our conversation today. Not necessary. Yeah, a lot of chromo terms in genetics.
I was lost a long time ago. I'm just going to try to catch up.
Okay, let's take a look at these 2 processes that the cell is doing. And these are really, really important. If you've had any of this conversation before, hopefully what I'm saying makes total sense to you. If this is brand new for you, my goal is to break it down in a way that makes sense. So the major thing that Watson and Crick figured out is that from DNA, from that double helical structure made up of ACTs and Gs. Somehow from that, proteins were made. And in their paper, they hypothesized correctly that those ACTs and Gs are being used to transcribe and then to translate the proteins. So we look at this little flow chart. You've got DNA, right, double-stranded molecule, and where is DNA found in the cell? In the nucleus. If you find DNA outside of the nucleus, that cell is dying, right? So DNA is in the nucleus. It is where it normally is found. And if you find it outside, it is a dying, it's a cell undergoing apoptosis. So we are going to undergo a process called transcription. Now, I'll talk to you more about this. And then through transcription, we're now going to make another molecule called mRNA, messenger RNA. This is an RNA molecule. It's made up of ACGs and Us. It has a particular job. It's called the messenger RNA. That messenger RNA is then going to undergo the process called translation. Translation is going to be the making of the proteins. Now, remind me, what organelle makes proteins? Ribosomes. And where are ribosomes found? Only out in the cytoplasm, right? So what does that tell us? Somehow, transcription happens where? In the nucleus, because that's where the DNA is, and the translation happens out in the cytoplasm. So what does that tell us about mRNA? It has to somehow get from the nucleus out into the cytoplasm. If you go back and look at, when you listen back, and you listen to the structure of the nucleus, you will hear me say that the nucleus has all over its surface nuclear pores, spaces, gaps, holes through which large molecules like mRNA can leave the nucleus and go out into the cytoplasm. Another thing, why do we need those big holes there? What's the nucleolus about again? Nucleolus was the organelle making ribosomes. Where are those ribosomes? In the cytoplasm. So again, we got big chunky things that are having to leave the nucleus and go out to the cytoplasm. So when you listen back and you listen to me talk about the nucleus, you'll hear me mention those nuclear pores. Very important, and I'll mention them again here. So we're going to go from DNA to RNA, and that is the process of transcription. Okay, let's just start walking through this a little bit. Let me give you a little backstory here. Of your chromosomes, 46 chromosomes, if I could spread out your little chromosomes into a long linear chain, your DNA in every cell would be about 6 feet long. It's pretty amazing when you think that 6 feet of a molecule are wrapped up inside a little microscopic cell in the nucleus. Of that 6 feet of DNA, only about 1.5% is necessary for making proteins. So if that was a 6-foot chain, only about 1 inch of the 6 feet are important for making proteins. The other 5 feet 11 inches of DNA are what were previously called junk. Right? It was the junk DNA. We really didn't know much about it. Or the other word for it was non-coding. Non-coding. It didn't code for DNA. Junk. Now, over the years, we have learned that there's a lot of important stuff in the junk. Okay? That the junk includes regulatory sequences and lots of other things. In that non-coding region, you'll also have introns, and I'll mention introns again in a moment, and a bunch of repetitive sequences. Turns out that my DNA and that of a primate are similar, and there are regions where there are like 10% of the DNA is exactly the same. So we know there's a lot of similarities, and some of these repeatable sequences are in our DNA. So this junk is repeatable little sections that go over and over and over and over without any understood meaning. So here is what the DNA kind of looks like in a cartoon representation. I'm going to draw in a little extra something here. Here is the DNA, double-stranded DNA. So there's my double-stranded DNA. Now from that double-stranded DNA, I'm going to make this mRNA. So this right here is mRNA. Now notice that it has interwoven yellow and purple sections. Those yellows are called exons. The purple sections are called the introns. If you look back on the slide before, I told you the introns are named for some of that junk. So all that purple stuff is going to be cut out. So all the purple stuff, all the introns are going to be cut out, and now what's going to be linked together are these yellow sections called the exons. And this is exactly what the cell does. It takes these pieces of coding. When I mentioned that the DNA was 6 feet long and only 1 inch of it codes for DNA, I didn't mean to suggest that the 1 inch is all over here on one side, right? That 1 inch is scattered throughout the entire genome. Little tiny, tiny little pieces of it everywhere. And somehow the cell has to find those useful pieces, get rid of the junk, and bring together all those useful pieces. That's kind of what's being shown here in this cartoon. So the junk is being cleaved out. It literally is lassoed out, just like you see here. And now that makes the final mRNA. So the mRNA only contains in it, the final mRNA only contains in it the useful sequences, the yellow exon sequences. Now, if I were naming this stuff, if I would have had a choice to be naming this stuff 50 years ago when they figured this stuff out, I would have said the introns stay in and the exons are going to exit. But they weren't listening to me. All right, so this really drives students crazy. The introns are you're going to get rid of the introns and you're going to keep the exons. Okay, it's weird how they name this. Oops. So I'm just telling you a backstory so that this all will fit together here in a moment. So if you look in a textbook from 75 or so, 40 years ago, it would say that there were about 100,000 genes in the human. And then as the years have gone on, the number of genes we thought were involved in our genome, that is all the genes in the human, has been going down. Then there was the Human Genome Project, right? And that's kind of news, old news now. The Human Genome Project, we finally sequenced all of the ACTs and Gs that make up the human, and we discovered that there's somewhere around 18,000 genes. We still don't even know exactly the number of genes in the human, but somewhere around 18,000. You'll hear between 18 and 25,000 still. Now, the reason that scientists started off long ago saying there were 100,000 genes is because there are about 100,000 proteins in you and me. So it was assumed that every protein had its own gene. Every gene made its own protein. That's what Watson and Crick said, right? They said that DNA, sequences of DNA, go on to make protein. So it was assumed that it was a one-to-one relationship. Now we know that's not true. We know that, for example, the cell can take this chunk, this would be a section of RNA, and notice we have exon 1 and exon 2, this is in orange, exon 3. And under different circumstances, we'll get rid of different pieces of junk and even get rid of some of the exons and bring together different combinations of the exons. So this would represent one gene over here, but this one gene can create two very different proteins, protein A and protein B. And it does so by linking together, just for example, here, exons 1, 2, and 4 were brought together. On the bottom, a totally different protein, sections 1, 3, and 5 were brought together. So what this shows us is this, that the genome, that is all the genes in your body, is smaller than your proteome. So we have about 100,000 proteins in your body. That's your proteome, all of the proteins. There's about 100,000 proteins, but there's only maybe 20 or so thousand genes. Okay? So the genome is smaller than the proteome. And again, like I said, back in the Watson and Crick days, that was not understood. They thought it was one gene made every protein in the body. Okay, so with all that little backstory put together, transcription is the process of taking DNA and making from it mRNA. So the template for this is the DNA, right? The starting molecule is the DNA. And this is going to happen in the nucleus. We've already said that DNA is found only in the nucleus in a normal healthy cell. So we know that's where transcription occurs. Here is a little cartoon representation of this process. We're in the nucleus, right? We have to be in the nucleus. We have this little cartoon representation of some DNA. Double helical. Remember I told you that the DNA is only held together by hydrogen bonds, so it can easily open up. We're missing all the cellular machinery here, but there are proteins and enzymes that kind of open up the DNA, unzip it. Molecules, again, that we can't see are going to be making a single-stranded mRNA molecule. Okay, so we're going from a double-stranded DNA molecule, and we're making, through the process of transcription, a single-stranded mRNA molecule. We're in the nucleus. Okay, so that's transcription. Now, once we've made that mRNA, that is carrying the message. Now we need to go about the business of making protein. So that is the second step here of translation. So really, making proteins is both parts. It's transcription followed by translation. They're both necessary for protein synthesis. The end of this process, we're going to make proteins. Now again, this is what Watson and Crick talked about. They said that DNA went to RNA, and RNA was then used to make protein. And that was coined the central dogma of molecular biology. That term is still being used, although we're finding out that it's a little bit more complicated than this, but that's okay. We'll think here in this more simplistic way for our introductory course. We've zoomed out. We're looking now at the whole story a little bit more. So again, there's my double-stranded DNA in the nucleus. Through the process of transcription, I just made a single-stranded RNA molecule. Now, what did I tell you was in the nuclear envelope? The nuclear membrane had some pores, spaces, gaps. And as you can physically see, the mRNA is going to squiggle through that nuclear pore. It's going to find its way. It's going to be guided into and go out of the nucleus. That mRNA is now going to be out in the cytoplasm. And here, there will be some ribosomes. Now, those ribosomes can be in two different places. They can be free floating around, or they could be attached to some endoplasmic reticulum. Here, the ER is not shown. We just see ribosomes floating around. There's one ribosome in this picture, and this is purple dude right here. So that's the ribosome. And that ribosome has the ability to take the message and make from it the protein. Here, the mRNA is shown in yellow, and the newly made protein is shown in green. That process of going from mRNA to protein is called translation. We're translating the message. Right? Translating the message. And then what do you see? At first, this protein molecule, it's a long chain of amino acids, isn't it? And what do we see in this picture? That protein is going to roll up into its own unique shape, right? And we would call that the tertiary structure. So it's going to go from a linear primary structure and roll up into a tertiary structure. So that's the whole picture, right, of DNA to mRNA to protein, or transcription to translation. If I say it enough times, it'll make sense to you. So here's the same thing. It just happens that the different cartoons use the same colors, just by chance. So we have the double-stranded DNA making yellow-colored single-stranded mRNA. Number one, therefore, is what process? Transcription. Now, these two words are in alphabetical order. Transcription does come before translation alphabetically, but you got to go about 6 or 7 letters before you see the difference. Now, number two is that time where what? The mRNA is going to go out through the nuclear pore. I won't have a word for that right now. And then number 3, we go from the mRNA and make the protein. That is, number 3 would be translation. Now, in order for translation to occur, what must be available out in the cytoplasm? Amino acids, right? There must be the building blocks, because we know that proteins are made of amino acids, so there better be amino acids out there. How many different amino acids are there? 20. So all 20 amino acids must be floating in your cells. Now, outside, I won't really talk about this too much, but if you look on your vitamin bottle, there are some amino acids that are in your vitamin bottle because they're considered essential. So some amino acids your body can make from glucose and other things that you take in. Some amino acids your body cannot make. And so to be healthy, right, you must have a balanced diet, have all 20 amino acids available, and as you're making proteins, all of those 20 amino acids must be available for you to make all the proteins in your cells. So that's just another cartoon of transcription and translation to give you an idea of the location. Here's some words just to put the whole story together. So translation is the making of proteins based on the information from the messenger RNA. It is the message. It's carrying the message from the DNA in the nucleus out to the ribosomes in the cytoplasm. It's of course happening outside of the nucleus, and the mRNA to do this process had to travel through those nuclear pores. The mRNA, number 5, is going to bind to ribosomes. Those ribosomes are going to be floating around in the cytoplasm, and there has to be amino acids out there as well. Now here's a new player that I haven't introduced yet, tRNA, transfer RNA. It's another type of RNA that will be important in making proteins, and I'll get to that in a moment. And then eventually we make that long chain of amino acids, which is the backbone or the core of the protein. So another way of thinking about this is that as we go from a gene to protein, we're going from DNA to mRNA to protein. Now what language, how many letters are in the language of DNA? A, C, T, and G. So if you think about it, it's a 4-letter alphabet, right? It's a 4-letter language. And then how about mRNA? A, C, G, and U. Now, there's a small change there. And this process is what's that process again? From DNA to RNA is transcription. What does that word mean to you, transcribe? Write down, right? Transcriptionist, to transcribe something is to copy it. So it's an almost exact copy, right? We're going from ACGs and Ts to ACGs and Us. We're not changing the alphabet. We're in the same language. We're just kind of writing it down. It's the message, right? You're going to transcribe the message and send the message somewhere. But then when we go from mRNA to protein, we're going from a 4-letter code, ACGs and Us, to a 20-letter code, or 20-letter alphabet, 20 different amino acids. So that's where we have to not just transcribe, but translate. So now you're basically changing languages. So we think about translation. You know, it's Rosetta Stone, right? You're changing the language altogether. So if that helps you think about how to practically think about these two terms, transcription and translation, that will help you when you're in the middle of the test and you're going, oh my goodness, I've got these all turned around right now. Now here's another picture of this transcription translation process, kind of a 3-dimensional representation. So the nucleus is shown here. The nucleus has all those little nuclear pores in it. Same old thing. There's the double-stranded DNA. It's undergoing transcription. It's coming, the mRNA is being made. It's coming out into the cytoplasm. Now this is a portion of that messenger RNA. And what you see is that there's blue, orange, green, and yellow. What do those represent? A, C, G, and U. Single-stranded. Right? And this big purple dude is the ribosome. And the tRNA is an important player here. These little colored balls out here represent the amino acids, 20 different colors and flavors out here. The tRNA is going to be carrying the correct amino acid to the ribosome. It's transferring the correct amino acid in. And what you see is that the tRNA has the opposite color. So here, green is matching up with yellow, and orange is matching up with blue, and the tRNA is reading or translating this message. So the tRNA is actually the critical cog here. Without the tRNAs working, this would not go well at all. Based upon the 3-letter sequence on the mRNA, the tRNA comes in and carries the correct amino acid in, and that is then linked together to make the protein. So that's sort of the big idea, the big story of transcription and translation. So if I take a look, the ribosome is made up of 2 parts. There's a big part and a little part, like an upside-down snowman here. The mRNA is going down the middle of it, and now this little tRNA is going to come in. So it turns out, I know it's hard to see, but this whole process is based upon groupings of 3. That's another thing that Watson and Crick figured out. In their paper, they also said, this whole thing is based upon 3s. 2 is not enough, 4 is too much. So 3 was the magic number, and they were right. So every 3 letters on the mRNA is called a codon. It codes for something. right? Every 3 letters is a codon. The tRNA that binds up to that carries on it 3 letters referred to as the anticodon. So the codon is on the mRNA, the anticodon is on the tRNA. They match up complementary. C's to G's and A's to C's to G's and A's to U, because we're dealing with a tRNA molecule and an mRNA molecule. They're both made of RNA. There's no T's here. So A matches up with U. Okay. And again, we're going to be leading through this thing in groups of 3. So this will be a good place to give you a little start, and then we'll pick up with this. Now, it's going to be a whole week before we pick up with this, right? Because on Wednesday, we'll have the test. That's all we'll do on Wednesday will be the exam, and I'll pick up this story with you the following Monday. We'll finish it up. But let me just kind of give you the take-home lesson, and then we'll dive back into the end of this next week. So in this picture, I've got my DNA. It's red. It happens to be red in all the pictures, it seems. This is only showing one strand of the DNA. The other strand, right, there'd be a C and a G and a T and a T, right? That's not shown. We got one strand. The DNA is now going to be transcribed to make mRNA. And what we have now are these 3-letter sequences. Each one's called a codon. And then from that codon, the tRNA is going to bring in the right amino acid. And it just happens to be that every time that the cell sees CGU, you're not going to memorize this. I'll show you how to do this. The tRNA is always going to bring in an amino acid called arginine, ARG. And every time the cell sees instead UCA, it's always going to bring in serine, SEER. And every time it sees UGG, it's always going to bring in TRIP. And ACU3. I'll show you how to do that. But that's the basics of this. So what happens then if there's a change in the DNA? If there's a mutation, then that mutation will be carried down through the mRNA. And if there's a problem with the mRNA, what could happen at the protein? The wrong amino acid could be put in. And if the wrong amino acid is in the place, then that means the protein may not be able to make its final structure properly. And as I mentioned in the lecture that you're going to review, if a protein doesn't have the right structure, then it may not do its job properly. So we'll come back and we'll talk about this. We'll break it down a little bit more detail. Now at this point, I know that between now and Wednesday, your focus is on chapters 1 and 2. But after Wednesday, I'd like you not to blow the weekend off, and I probably won't make this announcement during the test on Wednesday, but after Wednesday, don't wait till next Monday to pick up anatomy and physiology again. So you can certainly go into mastering after Wednesday, and you can start looking at chapter 3 homework on cells and organelles. You can certainly start moving forward with transcription and translation things. So please just don't blow the weekend off and then come back next Monday, completely cold. So that's my little goodbye to you. And I will see some of you in lab in a few minutes. And the rest of you I'll see on Wednesday at the exam.